Hi everyone, thank you for joining my podcast today. So our last episode covered the formation of the diploid zygote via fertilization in the fallopian tube. Next, the zygote is going to travel to the uterus where it will implant in the uterine wall for further development. However, it can take over a week for implantation to occur. The zygote is going to continue to develop as it makes its way to the uterus. The first stage of development that occurs after fertilization is called cleavage. The zygote is initially a one-celled structure, but we know that to become a functioning human, it needs many more cells. During cleavage, many rapid cell divisions occur through the process of mitosis. The first cleavage produces a 2-stage cell. The second cleavage produces a 4-cell stage, and so on. Once the zygote reaches the 16-cell stage, it is known as the morula, which is a solid ball of cells. So, we know that cleavage involves rapid cell division, but there are a couple more points to keep in mind. Since cleavage occurs super rapidly, there isn't much time for growth to occur before the cell divides. As such, the embryo divides into progressively smaller cells, and its total size remains approximately the same throughout cleavage. Another important idea to note is that cleavage typically occurs 12 to 24 hours after fertilization occurs. The embryo is still on its way to the uterus through the fallopian tubes at this point. There are many different types of cleavage, but MCAT test makers only require you to know about determinate versus indeterminate cleavage. Indeterminate cleavage means that the cells produced by cleavage are undetermined. They can still develop into complete organisms. Determinate cleavage means that the cells produced by cleavage are determined to become a particular cell type in the future. That is all of the content for this lesson about cleavage, but let's do a couple of practice questions that will help you to apply the material. The first question is, true or false? During cleavage, the nuclear to cytoplasmic ratio is decreasing. The answer to this question is false. In fact, the nuclear to cytoplasmic ratio is actually increasing during cleavage. This is because the cell is dividing into successively smaller cells that contain less cytoplasm, even though the genetic material in the nucleus is still dividing. And now for question 2. Are monozygotic twins, also known as identical twins, a result of determinate or indeterminate cleavage? The answer to this second question is indeterminate cleavage. This is because in cells that have indeterminate cleavage, these cells can still develop into complete organisms. So indeterminate cleavage is how monozygotic twins are formed during embryonic development. That is it for this lesson, but come back next time to learn about the next stage of development, which is called blastulation.